Section 19.7, Cofactors and Coenzymes. Some enzymes require an additional non-protein or what we call a prosthetic group in order to function. In this case, the protein portion is called the apoenzyme. The non-protein group is called the cofactor. Together, they form an active site called the holoenzyme. Cofactors can be metal ions, organic compounds, and sometimes they can be organometallic compounds, and must be bound to the enzyme to maintain the correct shape of the active site. Thus, these enzymes are only going to be active when the cofactor is bound to them. In this example, we see our apoenzyme, which is highlighted in blue, and then we see our substrate. Now you can see that the binding pocket of the apoenzyme, the topography doesn't match exactly with the outline or the conformation of our substrate. So what happens is we get no enzyme-substrate complex forming. But when the cofactor, in this case, it's a copper 2+ ion for this example, when the cofactor binds to the enzyme, now we have a functional enzyme with an active binding site. And the way they've done it here in the slide is quite clever. And that now that the cofactor has attached itself to the enzyme, the functional enzymes, you can see that the outline of the copper with the two ions, you can actually see that it matches the conformation of this green substrate. So now we get an enzyme-substrate complex and the reaction occurs. So this shows you that sometimes we have to have a cofactor in order to get a functional enzyme. Other enzymes require the temporary binding of what's called a coenzyme. Such binding is generally mediated by weak interactions like hydrogen bonds or hydrophobic interactions. Coenzymes are organic molecules that serve as carriers of electrons or chemical groups. In chemical reactions, they can either donate groups to the substrate or accept groups that are being removed from the substrate. If you take a look at this figure on the right-hand side, it shows a coenzyme accepting a functional group from one substrate and donating it to a second substrate in a reaction catalyzed by a transferase. So if we start with bullet point 1, you can see that substrate 1 is coming in and it has some kind of functional group attached to it. But the substrate 1 can't just transfer that functional group directly to substrate 2. We see that the enzyme has this cofactor attached to it. And then you see the binding pocket on the coenzyme has the same kind of round shape as that functional group. So what happens is that first substrate is going to bind to the enzyme, transfer the functional group to the coenzyme, and that's what's occurring here. And then the coenzyme is going to transfer that functional group over to substrate 2 in the third step. Finally, the products are released from the enzyme. Often coenzymes contain modified vitamins as part of their structure. A vitamin is an organic substance that is required in the diet in only small amounts. Of the water-soluble vitamins, only vitamin C has not been associated with a coenzyme. This table is a summary of some coenzymes in the water-soluble vitamins from which they are made. Coenzymes that will come up a lot in the future in this class are FAD, NAD+, NADP+, and of course, we'll see some others as well. NAD+ is a coenzyme that is of critical importance in the oxidation reactions of the cellular energy harvesting processes that we're going to see in Chapter 21. And the reduction occurs when NAD+ takes on a hydride ion, H-, to form NADH, which is the reduced form of NAD+, because you might remember that reduction in organic chemistry is the gain of hydrogens.